Hello students, welcome to BioSmart Academy. In today's lecture, we are going to discuss about Class II MHC molecule. Class II MHC molecule. Yesterday, I already taught you about the Class I MHC molecule. If you still not see that lecture, please first go and see Class I MHC molecule and then came here to watch this lecture. So, before I start this video, I just want to remind you that this channel is giving you a free module lectures of all units of CSIR NET syllabus. So please share this channel to all your friends who are preparing for this examination and also other biological science competitive examination, that is GATE, ICMR, DBT, all examination. This channel will be very beneficial to all the biological science student. So please share and subscribe my channel. So let's start Class II MHC molecule. First of all, we should know about the MSc2. As in the previous class, that is yesterday's class, we saw that Class I MHC molecules basically is present in all nucleated cell. Okay, present in all nucleated cell. Present in all nucleated cell. Okay, present in all nucleated cell. So please remember this point. Okay, and it basically related to TC. Okay, basically related to TC. Okay. Basically related to TC. Now, let's start Class 2. Basically, Class 2 MHC molecule is basically made up of alpha and beta, both chains. Here, alpha and beta chains both are present. Okay, Class 2 MHC molecule. In Class 2 MHC molecule, both alpha and beta chains are present. While in Class 1 MHC, only alpha chains are present and one beta 2 microglobulin is present, as I taught you yesterday, which is basically a invariant chain, which helps in association of Class I MHC with this cell membrane. Okay, that was about Class I MHC. But in Class II MHC, there are two types of chain present. One is alpha chain, one is beta chain. Okay, and both the chains have different molecular weight. Alpha chain have molecular weight of about 35 kDa, that is 35,000 Dalton, while beta chain have about 28 kDa, that is 28,000 Dalton. Okay, so basically MSc2 molecule is made up of alpha 1, alpha 2, beta 1 and beta 2. Okay, that is alpha and beta chain. Okay, and the main and major important point that I want to highlight you is that MSc class 2 molecules are member of Ig superfamily. Please remember this point, that MSc class 2 are the member of Ig superfamily. Ig superfamily. This may be asked in the examination and many times have been asked that which one of the following MSc molecules are the member of Ig superfamily. So MSc class 2 molecule are member of Ig superfamily. Clear? And in case of Class I MHC, we are very much familiar that CD8 basically there in Class I MHC, which is present in TC, is basically bind to the invariant chain. It is basically bind to the conserved sequence of alpha 3. In alpha 3, basically CD8 molecule basically bind. Okay, but in case of Class 2 MHC, the CD4 here, CD4 basically bind to the invariant portion of all MHC class 2 molecule. Okay, bind to the invariant portion of all class 2 MHC molecule. Clear? Now next, basically class 2 MHC basically generally express APCs. Okay, that is antigen presenting cell. It generally express MSc molecule constitutively on APC. How APC work, how the presentation is done by the APC through the MSc molecule, the whole detail of APC I will teach you in tomorrow's class, so that you get a clear idea about the processing and presentation of APC. Okay, so basically APC are classified into two types. One is professional APC and one is non-professional APC. Professional APC are those APC which are basically a major part of the innate immune system, that is NK cell, dendritic cell, macrophage. These all are known as professional APC. Lymphocytes are also example of professional APC. But in case of non-professional APC, is example are fibroblast cell, glial cell, and other thymic epithelial cells. These are example of non-professional APC, that is antigen presenting cell. And how antigen presenting cell, that is APCs, work, how it processed and present the antigen to TH or TC through MSC of both class 1 and class 2, we will see in the next class tomorrow. As in this class I totally focused you all in class 2 MSC. So these are some important points related to class 1 and class 2 MSc. Now next point. Some cell. See, one question may be asked in the examination that give an example of cell which express both MSc class 1 as well as MSc class 2 molecule. So basically B cells are those cells that constitutively express both MSc class 1 and MSc class 2. B cell are those cells that constitutively express MSc class 1 and class 2 molecule. Clear? And cell that display peptide associated with class 1 MSc, molecule to TC cell are referred as target cell, and cell that display peptide associated with class 2 MSc molecule to T helper cell are referred as APC. Okay, it means if we have clear this idea, it means class 1 MSc is TC cell, while class 2 MSc is TH cell. It is done by APC and it is target cell. But in many cases, in both the cases APC are used. In both class 1 as well as in class 2. So this is all about the class 2 MSc molecule. Now I make your concept more clear by draw a diagram of class 2 MSc molecule like I did it yesterday of class 1 MSc. See this diagram, you get all the concepts clear. Okay, see this diagram. Here I already told you that here 2 alpha chain and 2 beta chains are present and both have different molecular weight. But in case of class 1 MSc, only alpha chains are present with one invariant chain. Okay, so this is cytoplasmic part. Okay, and in case of class 2 MSc, both the chains are within the cytoplasmic tail. Okay, within the C terminal, within the cytoplasmic tail. Okay, and this is N terminal because this is a protein. And if I label this is alpha 1, this is alpha 2, this is beta 1, and this is beta 2. And MSc2 molecule basically belongs to IG superfamily. Please note it down. It belongs to IG superfamily. It belongs to IG superfamily. Okay. Now, in case of MSc1 molecule, we see that in alpha 3 CD8 molecule bind. But in case of this CD4 binding site is beta 2. This CD4 plus binding site is beta 2. Okay, and these two domain, that is alpha 1 beta 1, is the polymorphic domain. I already cleared your concept about the polymorphic yesterday. What is polymorphic? Basically, polymorphic is many alternative forms of the gene or allele that exists at each locus. It's polymorphic. Okay, so this is a diagrammatic representation of the class 2 MSc. Class 2 MHC. I think now you got your concepts clear about the MHC molecule, both Class 1 and Class 2. So this is all about the MHC molecule, Class 1 and Class 2. Okay. Now, before I complete this lecture, one important thing that I want to teach you is about the diversity of MSC molecule. Okay, diversity. Yesterday I taught you that the diversity of MSC molecule is due to the multiple allele. But there is also one reason, because of that also the diversity increases, and that reason is the presence of duplicated gene. So please note it down that duplicated gene is also one of the reason because of which diversity of MSC molecule occur. Okay, with similar function, but in many cases it includes that gene with similar but not identical structures and function may occur, and in this case the MSC may be said to be polygenic. Okay, poly means many, genic means genes. Okay, means many genes there. So this is all about the total concept of MSC, MSC1, MSC2. Clear? In tomorrow's class, I briefly discuss about the difference between Class 1 MSC and Class 2 MSC, difference between the Class 1 and Class 2, difference between the Class 1 and Class 2 MSC. In tomorrow class, I briefly discuss the difference between Class 1 and Class 2 MSC, because this difference is very, very important. So I discuss, and I also discuss about the antigen presentation and antigen processing. First processing, then presentation. How this was done by the APCs. I discuss it briefly in tomorrow's class so that your concept will get clear about the APCs. Okay, so meet you tomorrow. Till then, thank you.